Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 147th seminar, and I call it St. Jude Children's Research Hospital Day. I am in Rockville, Maryland, where the NIH Synthetic Biology meeting is currently held to discuss the future of synthetic biology after I gave a talk at J. Craig Venter Institute nearby yesterday, 2 days ago, the other location other than the La Jolla, California. It was great to meet many of my new people and my colleagues and friends here in East Coast. I am also looking forward to my short 2-week break without traveling until the Hawaii Synthetic Biology Young Speaker Series, this series in-person conference on December 9 to 12, my potentially last business trip this year, actually marking the record of more than 200 days of trips actually this year. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Madan Babu. He is a professor now at St. Jude Children's Research Hospital, United States, also the director of Center for Data-Driven Discovery and also interest in data science approaches to GPCR signaling systems. He used to be in UK, and I known him for more than a decade. I believe he visited a long time ago the Washington University in St. Louis many years ago. He is also an editor of Nucleic Acid Research, one of the Oxford University Press flagship journals. And I'm actually getting some email from him for reviewer requests, and sometimes almost always I probably accept the invitation because that's such a nice journal and such a nice paper coming in. Professor Babu, thanks so much for your great contributions to scientific community through your amazing research at the same time as an editor that's shaping the community, and the virtual podium is all yours now, and thank you again.
Thank you very much, Tay. Let me just share my screen. Yeah, thank you really very much for this kind and generous introduction, Tay, and good day, everyone. First of all, I really want to congratulate Tay for running this amazing series and bringing together so many outstanding, established, as well as like early career scientists together in this forum, and for enabling scientific dialogue and knowledge transfer. So this is really a great contribution to science and society, and I feel really honored to be part of this series. So as Tay mentioned, my name is Madan, and I work at St. Jude Children's Research Hospital. And before I started at St. Jude, I worked at the MRC Lab of Molecular Biology in Cambridge for nearly 15 years. So I currently direct the Center of Excellence for Data-Driven Discovery, and I'm also the Senior Vice President for Data Science at St. Jude. The next 5 minutes or so, I thought I will highlight about how we use data science approaches to make biological discoveries and utilize this knowledge for personalized medicine. So before I go ahead with the presentation, I had a brief exchange with Tay. I thought it'll be nice to spend a little bit of a slide providing an overview of my career history. So I did my undergraduate degree in biochemical engineering and biotechnology from India in Anna University, and obtained my PhD in computational genomics at the MRC Lab of Molecular Biology. I then did my postdoc at the National Institute of Health in evolutionary systems biology, and then I came back to the LMB to start my group as a tenure track investigator in 2006 until 2010, where I got tenured, and then continued to work there until 2020, which is when I moved to St. Jude Children's Hospital. Now, interestingly, we moved to St. Jude exactly in the middle of the pandemic. It was quite an interesting time, but I have to say we had such a great level of support from the organization to make this transition. All right, so technological advancements have really allowed us to generate huge amounts of information describing both biological entities as well as biological processes at different scales and resolution. These are the fundamental units for any systems biology or synthetic biology application. We need to understand the entities, we need to understand the processes, and then we could optimize them using many different types of methods and approaches. Now, all of this data basically presents very unique approaches now to computationally try to understand what are the new fundamental insights that can be gained, and how can we discover new scientific principles from these large volumes of data. And the current estimates, according to the European Bioinformatics Institute, is that we have access to over 150 petabytes of processed biological information. And given this deluge of information, what drives us scientifically is to find out how could we discover new insights and knowledge from these large volumes of data. So therefore, the mission of my group and my center is to develop data science approaches to make these discoveries and also reveal the general principles that drive living systems, with an emphasis on understanding human biology and specifically pediatric catastrophic diseases. So to achieve this objective, we investigate biological systems at three distinct levels of complexity: the atomic level, the cellular level, as well as the population level. And we bridge these different scales by trying to investigate how the impact of mutations pervade across all these different scales. And to this end, we develop interdisciplinary computational as well as experimental approaches that generates, integrates, mines, and analyzes disparate data sets. And these include data describing sequences, structures, expression, networks of interactions, and many more. So we have two major scientific directions in my group. The first one is on G protein coupled receptors, which are the largest family of human membrane proteins and are also the targets of over 1/3 of all FDA-approved drugs. So there are more than 800 genes that encode for GPCRs, and each one of these receptors govern almost virtually every aspect of human physiology, ranging from vision, taste, smell, as well as systems such as the cardiac system, the immune system, as well as the adrenergic system, and so on. The second focus area in our group is to try and understand what are called as intrinsically disordered regions in the human proteome. So if you take the proteome of any organism, there are parts of the proteome which fold into defined three-dimensional structures, and there are also parts that don't adopt a defined three-dimensional structures. And these are what are referred to as intrinsically disordered proteins. And my group is actively interested in understanding how exactly lack of structure contributes to function, or the ability to adopt multiple conformations contribute to the emergence of new function. In today's presentation, I'll try to highlight using GPCR work that we have done to describe how we go across these different scales. In the interest of time, I won't go into the details, but this slide illustrates the kind of insights that we have obtained by studying protein structures at the atomic and the molecular level. For instance, we have developed computational and experimental methods to investigate structures as networks and integrated them with genome-scale data sets to provide deep mechanistic insights into how GPCRs signal, all the way from ligand recognition to how the receptor is activated, to the selective binding of the receptor to the G protein, and more importantly, how binding of the receptor to the G protein results in GDP release and triggering downstream intracellular signaling pathways. Now, in a recent work, we also try to address a fundamental question. So people have been describing efficacy and potency of a ligand for over 100 years. But if you ask, what is the molecular origin of receptor efficacy and potency of ligand, that was a problem that was not deeply investigated. And we took an approach where we systematically mutated every residue in the receptor and measured efficacy and potency values. We looked at conformational changes that happen in the inactive state and the active state. And by systematically integrating these two large data sets, we revealed for the first time the allosteric network that drives the communication between ligand binding and G protein binding. And this provides us with very important insights that can be used to design small molecules that can tap into the allosteric network in rational ways that can then allow us to make more efficacious or more potent ligands, and at the same time minimize the undesired effects. It also revealed the presence of allosteric sites that are not exactly where the normal ligand binds, and that also means that we have opened up the space for druggable sites on these GPCRs. Now, at the tissue and the systems level, we also have done work where we integrated gene expression data with the GPCR information and showed how some GPCR genes give rise to a collection of isoforms with unique structural and signaling properties. We also showed that in different tissues, you have different combinations of isoforms that are expressed, and that can diversify the signaling response in the different human tissues. So the ligand will be the same, but you can have tissue-specific responses because different tissues express different combination of GPCR isoforms. So these findings basically implied that for these GPCRs, the observed signaling readout of a particular ligand may actually be a composite effect of the ligand acting on all the isoforms that are there. And given that isoform compositions can therefore differ between different tissues, activation by the same ligand could result in diverse system-specific signaling responses. Now, with these insights, in another study from our group, we integrated data on receptor structures with variant information from the genome sequences of more than 60,000 individuals in the human population. So this study, where we integrated polymorphism data in the human population with receptor sequence and structure data, allowed us to demonstrate that GPCRs that are targeted by FDA-approved drugs are highly variable at the drug binding pocket in the human population. And what this means is that individuals with polymorphisms in drug binding sites can either be non-responders or they could display adverse reaction to commonly approved drugs. So this study also, by integrating molecular data with population data, highlighted the implications for adverse drug response with implications for personalized medicine. So I hope this kind of gives an impression of how we investigate GPCR signaling at the atomic level, the tissue level, as well as the entire population level to gain general principles and make mechanistic discoveries. Now, I'd like to take the last minute to briefly mention the data science initiative that I'm currently leading at St. Jude, and would also warmly welcome conversations offline should this be of interest to anyone who listens to this video. So at St. Jude, we are generating data that spans an amazing spectrum of length and timescales. Now, realizing these opportunities, the data science initiative was proposed, and we have developed a comprehensive roadmap to turn this vision into reality. And one of the major focus of this initiative is to establish international relationship with organizations focusing on developing AI and ML methods, biological language models, as well as to bring quantum computing to address some fundamental problems at St. Jude. So if anyone's interested in these areas, I'll be very happy to engage in conversations in the future. With this, I'd like to acknowledge all my current and former members without whom none of this work would have been possible. I would also like to thank all the different funding agencies for the long-term support. Special thanks to St. Jude Children's Research Hospital, as well as the MRC Lab of Molecular Biology for funding my earlier work that I presented. Thank you all for your attention, and I'll be happy to answer any questions.
That is absolutely amazing. I do believe the future rely on fundamental understanding of basics, biology and science, and then apply that basic understanding to real translational application, and then you are doing both, I believe. And then absolutely also data-driven, research is future. I mean, that should be done more and more. And then we now have Nobel Prize winner this year about the AI or protein, design, etc. So future is bright with the young people like Brandon and also leader like you, Madan. So thank you so much. Absolutely amazing talk. I blown away. Thank you so much.
A real pleasure to be able to talk about this, and thanks for this platform to share some of the work that we do. Thank you, Jay. And I'm so excited about Brandon's work. I have to say, he's been such a real trailblazer in some of the work that he's been doing. I'm very much looking forward to your lecture, Brandon.
Okay, so now the main speaker of today with a longer introduction. Dr. Brandon DeKosky is an professor in the Department of Chemical Engineering at MIT and also core member of the Reagan Institute of MGH, Harvard, and then MIT. Research effort at his lab uh, has developed a suite of high throughput single cell platforms for comprehensive analysis of adaptive immunity. These efforts are advancing new approaches in biologic, drug discovery, and also for the comprehensive analysis of genetic and functional diversity in adaptive immune cells. I mean, immunity, now we probably, a lot of people understand because of pandemic we had, but immunology is not easy field to understand because I wanted to understand that one, when I was a master student more than 27 years ago. I bought a textbook and read half of that, and then I just stopped doing so because it's so complex system to understand. But I believe the Brandon is brave enough to tackle this very complex and but also challenging but very important field by doing the real research, at MIT right now. I believe I met him at a conference, and after listening to his amazing talk, I basically approached him and invited him to give a talk at this SynBYSS webinar series. Brandon, thank you so much for your time today and contribution to the society. And please take it away and all yours now. Thank you.
Great. Well, thank you again, Tay, for creating this forum for us to connect and share our work, and for having me here today. It's really an honor to talk about all the work that we're doing. And thank you so much, Madan, for the introduction and for sharing some of your work. Really excited, and I think you're absolutely right that collection of large scale biological and clinical data, along with sophisticated analysis of synthetic biology and clinically informed data is our way forward. So with that, I'll jump in. I'm going to talk today about some approaches that we've been taking for high throughput functional screening for immune-based drugs. So just background, I am going to try to set the stage for folks on the call about some aspects of our approach to adaptive immunity and the key things that we are trying to study. So of course we have antibodies and T cell receptors that are the dominant mechanisms that our adaptive immune system recognizes foreign proteins and foreign viruses and bacteria. So when it comes to antibodies, they're generated by the B cells in the body. B cells will encounter an antigen, and through getting help from T cells or other mechanisms, they'll become activated, and they'll either become a memory B cell and they'll be kind of waiting to see that antigen again, or they may become a long-lived plasma cell and they'll home to bone marrow and spleen, where they'll secrete antibodies for a very long time, sometimes the rest of the lifetime. And B cells do target external antigens. Each of us has over 10 to the 10th B cells, and each of those encode just a single antibody. So each one of us has this highly diverse, we refer to it as a repertoire of antibodies that are encoded by these B cells, and everyone is unique, even identical twins, because the B cell repertoire is generated throughout the course of life. We also encode T cell receptors, and T cell receptors are able to target internal proteins that the cell is expressing and then displays on what's called the major histocompatibility complex. So every cell has these peptides that are displayed on MHC proteins, And developing T cells have T cell receptors that specifically recognize that peptide as well as the MHC backbone. And T cells are tightly regulated, and the importance of understanding this interaction between T cells and these MHC peptides are essentially at the core of how we can suppress viruses or suppress cancers or understand autoimmunity. And so with these kind of enormous numbers, there's probably 10 to the 11th or more T cells in each person. So these are really, really high numbers of specific protein-protein interactions that we need to learn more about. When it comes to antibodies, antibodies have this Y-shaped structure. There's a heavy chain and a light chain. The heavy chain is shown in blue and the light chain is shown in yellow. And they recombine each individual antibody, and it has this binding site that's comprised of both the heavy and the light chain. There's these red loops in the center that are the specific binding sites. And if we can get the sequence of these antibodies, we can do high throughput data analysis, we can do in silico modeling, and potentially we could also screen them for reactivity to antigens. But we do need the heavy and the light chains to be paired together in order to do that. It's actually the same story with T cells. T cells have a similar structure. You can see a T cell in a co-crystal structure with an MHC peptide here. So that the MHC protein is shown here, and inside the binding groove is this peptide at the center. And the T cell receptor has an alpha chain and a beta chain, similar to antibodies, a two-chain structure. And you can see here in the zoomed in version how one of those loops from the alpha chain in the front and then the beta chain in the back actually really specifically interacts with that bound peptide. And then the other loops interact with the MHC protein backbone. And so it's really important if we want to study these T cell receptor interactions that we have to get the alpha and the beta paired together, and we need to do it in large numbers in order to understand the entire scope of an organism's immune response. So fortunately, we live in incredible times, and in most of our lifetimes, who's watching the video now, we've seen 5 orders of magnitude reduction in the cost of DNA sequencing. And there were a few really significant steps that have kind of brought us to where we are today. There actually hasn't been much change since 2017, so I'll update this slide when we have a substantial drop again. But you know, this ability to sequence DNA at such a relatively low cost has really transformed our study of immunology and immune receptors, because again, with 10 to the 10th, 10 to the 11th cells in an organism, that's really where we can start to sample. And so the traditional approaches have generally taken single cell RT-PCR and recently developed technologies like 10x genomics or other single cell sequencing to get single cell resolution. It has a relatively high cost and a low throughput, but it does work very well for smaller numbers to maybe medium numbers of cells. And traditional next-gen approaches look at just a single chain at one time, either the heavy or the light or the alpha or the beta. And it's much less expensive, it's much higher throughput, but you do lose the pairings, and so you lose that recognition of sequences. And so the question that we've been trying to answer over many years is how we can leverage these next generation sequencing technologies for paired immune receptor sequencing, and how to translate that NGS data into tangible functional insights about immune interactions. And so the first technology, this is actually where I started my graduate career using this technology and have kind of built on it through many years. The original technology was developed during my PhD at University of Texas at Austin with George Georgiou, and we developed essentially a workflow where you could isolate single B cells and capture them inside droplets. And using this approach, we could capture the mRNA from those cells, which contains both paired heavy and light chain DNA from that antibody. And using PCR, we were able to actually physically link the heavy and the light chains so that you could sequence them. And from there, we actually extended that further in work that bridged my PhD and my postdoc at National Institutes of Health at the Vaccine Research Center, and working with John Mascola and a number of other groups there, as well as with George Georgiou and the lab at Texas. We added restriction enzymes both to the center of the linker and then to the outside, and we developed a way to actually clone these in a natively paired way. So using this approach, which we started for antibodies, and we were able to get over 100,000 unique antibody genes from a single individual, or as I'll talk about later, we've applied for T-cell receptors for over 40,000 natively paired alpha and beta distinct clones from a single individual. And so we're starting to get sampling of immune repertoires on scales that are relevant for answering some really important biological questions. So I'll skip through a lot of the details about the synthetic biology of how we made this construct. But just a couple of the highlights, we at the center of the linker, we clone a bidirectional galactose-inducible promoter, Gal1, Gal10, so that when you feed yeast galactose in this yeast display plasmid, when you feed yeast galactose, they start to express the proteins. And then in one direction, we have the heavy chain along with the classic AGA1, AGA2 yeast display anchoring, surface anchoring system. And in the other direction, we have the light chain. And there's a two-step restriction enzyme cloning process that essentially transfers those natively paired antibodies into the display shown here. So we're displaying native Fabs on yeast. We've applied this technology for natively paired screening to a few different questions and a few different problems. We developed actually an approach to screen antibodies based not just on binding alone, but based on affinity. And so here in the top right, you can see screening a population of antibodies targeting HIV from a donor that had a very strong response to the HIV fusion peptide, or FP. And we were actually able to gate those antibodies based on their the ratio of display to antigen binding, and essentially using next generation sequencing, fractionate them between low and high affinity. We extended that approach to understand reactivities and cross-reactivities in flaviviruses with Zika virus and yellow fever virus, including a couple different forms of the maturation state. And we also applied this approach for SARS-CoV-2 to try to understand what were the sequence-based and structural-based features that led to potent neutralization at the receptor binding site. So that was really exciting, building these technologies for paired heavy and light chain antibody sequencing and screening, which we're still using today in a number of contexts. Another question that we started to get interested about was, if an antibody is pretty good, but it's not quite where we want it to be against a pathogen like HIV or malaria, can we start to mutate it in targeted ways and collect, genetic, functional, and structural data to understand how we can improve recognition? And so a postdoc in the group, Bharat Madan, really took on this problem, and we established a site saturation mutagenesis approach where we would essentially replace each amino acid, one amino acid at a time, with an NNK codon that would encode for all possible amino acids at that site. And so we would clone these single saturation mutagenesis libraries into yeast display. We use that affinity gating strategy to understand which mutations would lead to low, medium, or high affinity, and next generation sequencing to read out the results. So this is just a sequence map of essentially what each of these single mutations look like and their effect on affinity. This is for an anti-fusion peptide antibody called VFP16.02. And it's for a single antigen known as the BG505 SOSIP trimer. It's a stabilized trimer that's well characterized for HIV. It's a particular strain of HIV. And So not to go into too much detail, but in this case, it turns out that about 40% of the analyzed mutations were deleterious, 60% provided little to no change, and just 1% enhanced affinity to the fusion peptide. You'll see that a lot of the mutations in the CDR3s are deleterious. This antibody was also, potentially pretty well optimized at the CDR3. It turned out that some of the best mutations for this antibody were actually in the framework 2 region, so not the extended loops, but the regions that are essentially keeping the structural folds of the antibody. And in this case, it seemed to align the two heavy and light chain domains in kind of just the right way to improve neutralization. So extending this approach, another graduate student, Bailey Bonnock, took on an extension of this type of work where we took the best known anti-fusion peptide antibodies, or one of them, and we started to combine mutations. So we did the site saturation mutagenesis, and then we started to combine mutations and screen against a number of different stabilized probes to try to cover the breadth of HIV as much as we could. Interestingly enough, we did one round of this mutagenesis, and we found that and screening, and we found that a particular variant of the fusion peptide that has an insertion, we call it variant 4, right here. So there's a 1 amino acid insertion. We were really not picking up a lot of neutralization, only it was neutralizing 11% of viruses that had that fusion peptide sequence. So we did another round of multi-mutation screens to try to pick that up as well. This is just a plot of the yeast display libraries as we were going through single mutant screening. You can see that some of the libraries with single mutations enriched against certain HIV probes. For example, this one enriched very nicely, whereas others there wasn't always a single mutation that would improve binding to that particular epitope. And then when we went to multi-mutations, we were getting more consistent improvement, from combining enriched mutations together and rescreening, including, as I mentioned, against that variant 4 of the fusion peptide that has a 1 amino acid insertion. So you can see the pre-sort library in blue compared to the sorted libraries in red, or the wild type library in blue compared to sorted libraries in red. And you could see that we picked up binding to this particular antigen that wasn't there before. And so when we started to look in both predictive panels and then the larger 208 virus panel, there was one particular mutation that was extremely promising. It actually turned out that it was the most broad and potent anti-HIV-1 FP antibody ever reported. This is among antibodies that specifically recognize that fusion peptide as its dominant epitope. And we're really excited because if you look at clinical candidate antibodies that have been discovered in terms of their geomean IC50, so this is like a correlate for how potent an antibody is versus the breadth of how many strains they recognize. This actually brings the fusion peptide epitope into the range of clinical candidates. Most of the antibodies that are in this pink shaded box have been included in one clinical trial, either for prevention or therapy. So we're really excited that this actually brings this epitope into that range where it could potentially be useful at the clinic. Interestingly, there were 3 substitutions that led to this. One was just at E2K, so this is just the second residue of the heavy chain. And it turns out that it forms this salt bridge with an E87 that's relatively conserved on the HIV trimer. That's not one that you would necessarily have looked at. Often people are looking at the binding sites, and this one was much farther away. We did have a traditional binding site mutation as well, this T58F. It moves tyrosine 94 just a little bit. That makes contacts with the bound fusion peptide. That the antibody forms a groove that the fusion peptide is captured into, and this adjusts those contacts in a little bit more traditional way. And the last mutation that was very interesting was this A33P mutation that essentially broadens the binding groove. And so you can see the heavy chain and the light chain, and then the fusion peptide in purple. And this A33P actually makes a little bit more space to accommodate insertions that do happen in diversity of HIV strains. And so this kind of gave us a little bit of a functional map of, if you're going to improve antibodies that recognize this fusion peptide epitope, or you're trying to design vaccines to elicit it, this gives a little bit of a structural map of how that would be accomplished. So I mentioned we're also interested in understanding T-cell receptor activity against viruses and cancers. And T-cell receptors are very complex to study compared to antibodies because the energy of T-cell receptor peptide MHC interactions is much lower. So the affinities are much lower. And for antibodies, it's kind of like, you have an on relationship. It can be, picomolar interactions, very, high, there's a lot of energy in that bound state that leads to high affinity. For T-cell receptors, the difference between an on and an off reaction is much lower and the affinities are much weaker. And so they're a little bit harder to study both computationally and experimentally. So trying to address this, two former postdocs in the group, Ahmed Fahad and Andrew Chung, developed this approach where they would actually capture, again, single cells, capture the mRNA, and pair the alpha and beta chains of T-cell receptors and clone those into lentivirus. So lentiviral display of the natively paired alphas and betas. And then we could screen both with peptide MHC tetramers. The tetramer gets over the low affinity of interactions. Or we can also screen with cell-based display to understand activation potency or activation potential and no need to make tetramers. So just sharing a little bit of data from these studies. Screening with peptide MHC, the response can be a little bit rare. This is in a infectious mononucleosis patient where there was, some predicted epitopes. And the library is enriched quite well. You can see a couple of different populations from multiple TCRs that are in that sorted library. And other libraries may be a little bit weaker enrichment, but enriched very nicely after 2 rounds. And so we can sequence these sorted populations and understand the identity. And those alpha beta TCRs are also immortalized. So we can go back with any number of peptide MHCs or activation-based screens in order to identify the reactivities to many different antigens. So we're also trying to look at cell-based activation. We can clone T-cell receptors into native human T cells to look at native function. And we can also do activation-based cell sorting to try to select directly for cell killing or activating T-cell receptors. And so in just some recent work, we are looking at functional detection of anti-cancer TCRs. And this is some data from a humanized mouse and a humanized mouse model where the mouse were given a cancer cell line and checkpoint inhibitor therapy, and we clone those libraries. And in screening the libraries against the co-cultured cells, we can actually detect an increase in TCR reactivity. And we're following up on these screens to identify the specific TCRs that are being activated against specifically those cancer cells. That's really exciting because we don't necessarily have to know the peptide MHC. We don't have to know what the antigen is. We can identify T-cell receptors that target cancer cells directly, and then from there find the antigens. So a really exciting approach, and we have a number of parallel experiments going with human samples and human models as well. So the last approach I want to talk to you about is trying to understand not just the affinity-based functions, but also some of the beyond affinity functions that antibodies can do. So a lot of antibody discovery is based on the affinity of binding. So whether an antibody binds to an antigen. But when it comes to antivirus antibodies, there's actually the critical property is often neutralization. So whether an antibody will block viral infection in vitro. So Unfortunately, it's an analytical assay. It's a multi-day well plate assay where you have 10,000 cells in each well, and you have a series of dilutions. And it's pretty low throughput, but it's a very important assay, and this is the critical feature of whether an antibody can essentially suppress a virus in the body. Unfortunately, the proxies are poor substitutes, so binding to the extracellular domain of a virus is not necessarily going to lead to protection, and in many cases it does not. A similar problem with kind of beyond affinity function is with G protein coupled receptors or anti-membrane antibodies. Madan gave a great introduction to GPCRs, so no need to repeat, but for producing these proteins, the membrane domains are often essential. You can't even make an extracellular domain for most GPCRs. And the analytical assays are actually more complex than ELISA. And even when we do have antibodies, they're mostly antagonists. They're very rarely agonists. And so we've been trying to address these issues by finding ways to go from the traditional 10,000 cells per well as one assay and a plate-based readout and move them into droplets so that one cell per droplet is one assay. We can have 1 million assays in a single microtube, a computational readout of the data, testing millions of clones at a time, and use directed evolution based on function rather than affinity. So we've actually been really excited to be developing this technology for neutralization screening. So the way our approach works is we take single cells that are infected, that have a viral entry receptor, so they can be infected with a pseudovirus reporter gene. And we transduce a library of antibodies into these cells and emulsify them into single droplets. So each cell is inside a droplet, and inside the droplet, it secretes its own unique antibody that can be ready to be assayed in a functional context. It's, the soluble antibody in essentially the final druggable format if you wanted to use this antibody as a drug, or if you're trying to understand its behavior within the body. So still inside those droplets, we use pico injection to merge these droplets with other cell-containing droplets. And inside those droplets, there's two things that could happen. One would be that the antibody is neutralizing, and so the pseudovirus will not be able to infect. In the other case, the antibody may be non-neutralizing or not targeting, and the pseudovirus will infect, and the cell will light up, in this case, with a GFP reporter gene. From there, we can actually recover the cells, and instead of reading out color like an ELISA well plate, we sequence the entire populations of cells in both of those functional sort groups. And by reading the data and looking at the differences in clones that go into the infected and non-infected groups, we can actually identify which of those antibodies have neutralization function. And then, of course, we can make a small number of these antibodies, test them, validate them, and see where the structural interactions might be occurring. So here's an example with a synthetic library with HIV-1. Just going through the data pretty quickly, we have predominantly non-neutralizing antibodies shown in black. And then there's a couple of clones that are mixed in at about 10% VRCL1 and VRC34 that do neutralize HIV. So if you look at the non-infected population, actually a majority of cells don't get infected in these experiments. And so you don't see all of that much movement because most cells in the droplets are not challenged. But the throughput is high enough that a substantial number are. And in those infected cells, you actually see a near total loss of neutralizing antibodies. And so by comparing the two populations, we can actually see a 300 to 1,000-fold difference in the prevalence of neutralizing antibodies across the two populations, which is a really strong signal and very easy to read bioinformatically. So building on this work, we've actually started to look at neutralization of site saturation mutagenesis libraries, kind of like we showed before with affinity, but now screening based directly on neutralization. So we screened a site saturation mutagenesis library against both the D614G ancestral strain of SARS-CoV-2 and also against XBB.1.16. And collected the data. We had a number of variants that were detected in NGS for their neutralization signals, and we selected based on those a few that really seemed to have strong neutralization signals. And it turns out that of those, 5 out of 5 that we picked for expression actually did neutralize single variants, and of those, one of those single mutations actually was 5-fold improved. We looked at the structures of these, and it was really interesting. A couple of them were in the CDR3 or close to it, some kind of more common types of sites for improvement. And then a few of them, including the very best of them, this S17M, were pretty far away. S17M is, it's up here, it's way out in space, and it's probably mediating some kind of allosteric or long-range interactions that you really wouldn't predict based on, the known crystal structure of this antibody. So with that, we've successfully transitioned these functional assays into droplets. We're doing millions of functional tests in parallel, and we can screen, native forms or synthetically engineered libraries. And we're really excited to use this to screen biology in its native context, without necessarily needing to modify probes in order to study the important aspects of antibody function. So with that, a lot of people that I want to thank, starting with my lab and the folks who were doing, all of this work, Bharat Madan, Bailey Bonac, Andrew Chung, Ahmed Fahad, Shaoli Pan, Matias Gutierrez, and Mateus Oliveira de Souza, who really took the lead on a lot of the work that we were doing today. Of course, UT Austin, where my work in this area started with George Georgiou. My colleagues at Vaccine Research Center, where I did my postdoc, including John Mascola, Peter Kwong, Nancy Solon, Danny Dewick, and Bob Cedar. And of course, all of our collaborators on this work and our funding. And many thanks to you all for taking the time to watch this seminar. And I look forward to being in touch with you all soon. Thank you.
Fantastic talk. I forgot to mention during your introduction, I almost joined Giorgio lab for my postdoc, but somehow my wife wanted to live in San Francisco. That's why I joined the Chris Voigt lab at the time. Professor Madan Babu, do you have any question, comment before I go to the another?
Yes, I mean, both comment and a question. So Brandon, really phenomenal work. It's wonderful to see, the kind of problems and how large scale approaches that generates new data and analysis of this data in creative ways also informs of, the most potential candidates that you can screen from very large combinations of possibilities. The B cell work was brilliant, and then this pairing was a real challenge. And I think it was very nice that you highlighted, approaches where you can do this in a systematic way. And then this encapsulation to discover neutralizing antibodies is another major step. So I had one question about the pairing. So you kind of went over, presumably in the interest of time, the pairing between the alpha and the beta chain. How exactly do you ensure that pairing happens? Is it like the droplet ensures that, all the contents of the mRNA remain in the droplet, and then you do some sort of a targeted ligation, and then you would get that out of cells, and then create the library? And what's the efficiency of that process, and how do you ensure that there's no leakage and perhaps mispairing from those steps in your experiments? So that was one somewhat specific question that I had, and I'll go back to a broader point later.
Yeah, that's a great question. And you're right, in the interest of time and to focus on, various aspects, I didn't go into too much detail. Also because it's been published, and so I didn't want to go into too much of that detail. But the way it works is, you're exactly right, where we have a droplet that is essentially isolates the alpha and beta mRNA from a single cell. And, of course, alpha and beta are actually expressed on different chromosomes. They're expressed on different mRNA strands. They're different polypeptides, and they only get packaged at the assembly level. And so there's, when it comes to the genetic DNA, there's the only association is that they're within the same cell. They're not physically linked. And so what we do is we use magnetic beads as a shuttle that captures all the transcriptome from all the polyadenylated mRNA transcripts from a single cell. We use that as a shuttle to bring it into an emulsion reverse transcription overlap extension PCR, and that actually physically links the alpha and beta genes. Then we can break the emulsion and do nested PCR to purify and then, of course, clone using restriction enzymes and things like that. So,
Got it.
yeah. So in terms of how high fidelity is it, high throughput biology, doing, 1 million cells at a time is, of course, not as high fidelity as doing, 1 cell at a time. But it's still pretty good. I would say that probably 90% or greater of the transcripts are natively paired. And it's very easy to see in the data signals which are the native pairs and which are not, because the native pairs are at such a high prevalence. And so you can read that out fairly simply. And from single individuals, we've collected over 40,000 unique T cell receptor clones, which is I think about as many as most folks do see when they do other types of studies. So we count a little bit differently because we like to cluster the CDR3s and the V genes to make sure that we're not counting like sequence errors and things like that. Yeah, overall it's pretty good. It's hard to get exact counts and things like that, but it works well enough for high throughput screening libraries.
Yeah, thank you. I'll probably pass on for a question that we have from the audience, and maybe I can come back if that's okay.
Sure. Let's address this question from Kenyon. So question is, within the Droplet assay, how do you get the cells that secrete non-neutralizing antibodies to express GFP?
That's a great question. So the inside the droplet, it's the pseudovirus that encodes the reporter. And so if a cell is expressing a non-neutralizing antibody, the pseudovirus will be able to infect the cell, and then that will lead to the reporter gene. Different viral systems have different mechanisms. Sometimes the reporter's in the virus encoded, like a gene encoded in the pseudovirus. And sometimes there's a gene in the cell line that will activate a reporter when the virus infects. But generally, the expression is linked to that pseudovirus infection event. Yeah.
Okay, so Dr. Babu, go ahead.
Yeah, great. Maybe I have, if you don't mind, one general question and one more forward-looking question. So you highlighted, in your talk that you, without having the need to know what the epitope is or what the cancer cell specific peptide sequence is, you can discover the relevant PCR pairs that might be doing the job. But I was just wondering, what is the state of the art in being able to link specific TCR pairs to the relevant peptides, Brandon?
Yeah, so just off the cuff, this is an area that is developing so rapidly and so quickly. There are a number of groups and a number of approaches to link TCR and peptide MHC pairs. There's some library versus library screening approaches that have been developed or are being developed. If you can do peptide MHC libraries displayed on yeast, for example, like Chris Garcia and Michael Birnbaum's lab do. You can, of course, do combinatorial peptide pulses with a known TCR. And there's also ways of, of course, screening. If you know the peptide MHC, you can try to select for different TCRs. So I think it's an area that's going to see really tremendous growth coming up in these next few years. It's certainly still a challenge, but I'm excited to
You
see
did, yeah.
how we
Yeah.
how we continue to find good ways to address this problem with good scale. Yeah.
Yeah, because if we can crack the TCR peptide code, that's a pretty big step, isn't it, in our
Yes.
ability to harness the immune system for directed applications. The one forward-looking question that I had, you beautifully highlighted, the data coming from single site mutagenesis to discover these broad recognition antibodies and also these hotspots that you wouldn't have been able to predict from sequence alone. But with all these developments that are happening in protein language models and perhaps like, structure prediction methods and methods to predict protein-protein interactions, this is also another active area like what you described, which is, changing so quickly. Are these directions that you are pursuing yourself or through collaborations that, you're trying to use your own data to at least train them and learn them for predicting the combinatorial mutagenesis that might do the job? Because the space just becomes higher and higher when you go from single mutations to pairwise or triplets of mutations, and they can also have some really interesting sequences that can do the job. Any thoughts there would be great, Brandon.
Yeah, absolutely. I think we live in amazing times. The capabilities of computation and machine learning have exceeded anything I thought I would see at this time in my life. And I think that those are underpinned by, of course, important develops in algorithms, but also they require extensive and well organized, complete, rich data sets in order to function very well. And that's really where I see my contribution as being the highest is in using these experimental platforms to
Data
develop
set.
really rich
Yes.
data, complex data sets that can be mined with all of these amazing tools that we have now. So
Great.
yeah, we've been focused mostly on the experimental data collection as
I mean,
a
good
way of
data set
advancing
matters for any of the training and any development of novel algorithms. I think there was one more question from Vikas on the Q&A. Yes.
Okay, so question from Vikas, how technically challenging it is to set up droplet-based sort?
Yeah, so that's a great question. I would say one of the most important variables is background and time and interest. So it can be for somebody who has a lot of experience in microfluidics and molecular biology. I'd say it's not so difficult, and depends on the background and skills that someone has. Feel free to reach out to me if you have an interest in learning or setting up or working with these tools, and yeah, we'd be happy to discuss more.
Okay, Madan, do you have any other question or
No, I
comment?
mean, other than like, a generic comment to say thanks to Tay for, bringing people who wouldn't probably normally not cross paths, but nevertheless, whose work can have huge impact on each other's science and the way we think. And I have to say today's pairing that you did is an excellent example because I was very inspired by Brandon's presentation and the way he was thinking about how he uses combinatorial libraries to address some very important fundamental questions. And I also hope that there'll be more opportunities for further interactions with Brandon as well as us, Tay. Congratulations on putting together your 147th, lecture series in this on this topic. Thanks
So,
again, Tay.
so thank you so much. It's my great honor to have both of you today. It's all thanks to all the speakers and audience showing their excitement and contribution to the community. And I probably need to do that more than decade. The first seminar, Francis Arnold actually said I should do that for minimum of decade. So I continue to recruit people to
Yeah.
speak for this
And
seminar.
it's a great way to record knowledge,
Yeah.
and people can always go back and see where the state of the art was.
Excellent.
And I think that's another very interesting way to know about the history of the field. I mean, the introduction that he gave, Brandon, was excellent because it defines the problem statement so clearly that any young early career scientist who is going to embark on this journey of wanting to solve an important problem can be very clearly inspired rather than broadly inspired too.
Sure.
You know, so I think that's very cool.
Thank
Thanks
you.
again. Thank you, Brandon.
If you're available, please join us in Hawaii conference
No,
December.
you know, Dr. Vikas Trivedi, who's one of the attendees, is from my group.
Oh,
You
good,
know,
good.
so Vikas is a synthetic biologist more
Uh-huh.
broadly. So, you know, I'll probably go and brainstorm with him to see
Sure.
if one of us would be able to join this meeting in Hawaii that you
Yeah.
mentioned.
Absolutely. So the same to same to you. I mean, if you have time, but this is very not many time left in terms of booking everything. So we only have a few weeks left. But still, if
There's
you're
a one.
interested, yeah, this is gonna be the December 9 to 12 in Hawaii.
Actually,
But the good thing is that the price for trip is not that
expensive.
expensive compared to other season. I would say from the East Coast less than $600 or
So we can figure it out.
something
Yeah.
like the flight, and then hotel is something like I got the 170 per night.
Got it.
Compared to Hawaii other time, it's kind of relatively cheap.
Yes, I can imagine. Yes.
Okay.
Great.
So let's close. Thank you all for joining and staying today. We'll meet again on December 5, Thursday, the same time, the same Zoom link. Note we will not have the webinar next week, of course, due to Thanksgiving holiday in the United States. We'll have the Professor Sai ready at ETH Zurich. I believe he is also from the Joji Jojo lab, if I remember correctly. And
His Sai and I overlapped, and he was one of the first people teaching me, molecular biology tools
oh my
when I
goodness, what
started.
a coincidence.
Yes, yeah.
And
So
very
yeah,
similar, right, on T cells. Like, yeah, it's going to be very exciting. Yeah.
yeah, very exciting talk, I believe. So I met also him from one of the conference, I believe the Gordon conference. I probably met him again. I knew him for many years. And then
Great.
another speaker will be Professor Elizabeth Wayne, now at University of Washington. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I stop recording. Just give me 1 second.